Before we hand over to Julian to deliver the oration, it's nice to recognise the history of people in the ASM. So I'd like to tell you a little bit about Sid Rubbo, who the award is named after. So Sidney Rubbo was born in 1911 in Sydney, and I'm going to have to use this clicker again. There we go. So he didn't live for a very long time, and that was very sad, and you'll understand why that's sad when I tell you about what he did for microbiology in Australia. So he graduated from a science pharmacy course at the University of Sydney and moved to London and obtained a diploma of bacteriology and a PhD. And he was appointed as a senior lecturer in the bacteriology department at the University of Melbourne in 1937. And at that time he taught, he did a lot of teaching, and he taught students of medicine, dentistry, science and agricultural science. And so he taught at the same time he studied for a medical degree. So he taught and studied at the same time. And he did that successfully. He did both of those roles very successfully. And so he negotiated that dual role of teacher and student very well. And after completing a year's residency at the Royal Melbourne Hospital, he returned to the department in 1944, and in the following year he was appointed a professor of bacteriology in 1945. So why do we recognise him, and what did he do during his career? Well, he built a first-class teaching and research department at the University of Melbourne, and he's very well known for having done that. He was especially interested in the area of sterilisation and disinfection, and he wrote a very famous book about all of that. And he discovered a disinfectant as well, which was aminacrene, and it was said to be the first Australian drug recognised by the British Pharmacopoeia at the time. But he did a lot more than that. So he also worked on the prevention of tetanus, the reduction of cross-infection in hospitals, and even the sterilisation of spacecraft to ensure that terrestrial organisms didn't contaminate outer space, which was, very, very forward-thinking. So he's best remembered as a brilliant and provocative lecturer who inspired a generation of students, and we hear a lot about him, that he was very inspirational and that he spent a lot of time with his students, inspiring them as well. Before I get to this, I just want to also say, because it's really interesting to understand people outside of the science that they do. So he's also known as being very outspoken on political and moral issues, and Rubbo opposed Australia's involvement in the Vietnam War and campaigned with authority against the use of biological and chemical agents in warfare. So I think that's something to really note about him. He was also interested in the arts, especially in painting, sculpture and the theatre, and his pastimes included tennis, woodwork, winemaking, it's Italian background, and playwriting. So he was one of the founders of ASM, and so he chaired the inaugural meeting of ASM in 1959, and he was ASM's second president. So I think, I've heard a lot about him. There's all folklore about Rubbo, and of course I never met him, but the thing that I take away when I hear stories about him is that he's remembered with a lot of affection and a lot of respect. And the respect that he had then is even higher now for what he built for the society. And so we've named this award after him, and every year we give out this award. Every second year we give it to an international person, and then we give it to a national person as well. And so I can say that in the same way that Rubbo is thought of with affection and respect, I think today's Rubbo Oration winner is also thought of in the same way. So let me say a few words about Julian. So Julian obtained his PhD in, I don't think I've got another slide, I'm just going to check. Oh, sorry, I've got this other one. So the Rubbo Trust was established with money raised, sorry about that, after Rubbo's death in 1969. So he died a very young man. It was very sad. Contributions were received from colleagues and friends across many, many circles, including the artistic circles that he really supported. The Trust allocates funds for an abstract sculpture and has retained funds to endow the Rubbo Oration, which we give every year, as I just said. And so let me go back to Julian now. So Julian obtained his PhD in the Department of Microbiology at University of Melbourne, fittingly. And after taking up postdoctoral positions at the University of Wisconsin and ANU, he took up his first academic position at Murdoch University in Perth. He then moved back to Melbourne and took up a position in the Department of Microbiology at Monash University, which he has been there until now. And his research is focused on genetics and pathogenesis of pathogenic Clostridia, and that's what his subject will be today, and he's going to tell you about that. He's a fellow of the American Academy of Microbiology and a fellow and life member of the Australian Society for Microbiology, and he actually served as our president from 2004 to 2006, and he still works for the Society and is chair of our NEQB board. So what else can I tell you about Julian? Because I've worked with Julian for a very long time. So I was a postdoc in Julian's lab for 17 years, Julian. It was a long time. There was mostly good, but there was some, we did have very robust discussions. There are a lot of people in this room who worked with Julian, and I know that there's Priscilla Johansson was a PhD student in Julian's lab. Sheena McGowan was a PhD student in Julian's lab. I'm going to forget people here. Well, I worked with him for a long time. Hayley Newton studied with him. Sasha's here as well. Sasha Pitto studied with him. I'm sure I've missed people. Lots of people who've worked with Julian. And the one thing that I can say about Julian, his science is great. You're going to hear about that. But he was always a very welcoming and person, and he always invited us into his home, and we got to know his family, and not everyone does that, so that was a very generous thing to do. But I particularly remember, this speaks to Julian's character, I particularly remember one year Priscilla, she's here somewhere, Priscilla was a PhD student, and we had a party at Julian's house, and Priscilla brought along a piñata, which was great. It was a lot of fun. But Julian decided to hang it in the house instead of outside. And then when we needed a stick to find a stick, Julian said, I've got a hockey stick. So he brought out a hockey stick for us to whack this piñata with. And you're blindfolded when you're hitting the piñata. So it's amazing that no one ended up with broken arms or broken heads, because that hockey stick was being swung very vigorously. And then when someone cracked open the piñata, Priscilla had loaded it not just with chocolates, but with glitter. So there was glitter all over Julian's house, and actually I remember even 2 or 3 years later Julian coming in and saying, I'm still getting glitter out of the carpet. And so that was the gift that it kept on giving. But my point with that story, as funny as it is to say Julian always invited us into his house. We got to know his family very, very well. And he didn't get cross with the glitter incident. He never got cross. He just laughed. And so it was a really great environment to be in, and he was always very welcoming that way. And so I think it's great to have his partner, Cheryl, here with us today. So thank you for coming along today. The thing I'll leave you with before we get to Julian talking to you is to say that I think Julian's biggest contribution, actually, we talk about research all the time, but I think his biggest contribution to microbiology in Australia is his teaching, because he has taught thousands of students microbiology in this country. And so we need to recognise that too. So it's a delight to confer the Rubbo Award on Julian, and I'd like to invite him up to give the oration.
Thanks, Dina. I couldn't think of anyone that I'd rather have introduce me to give any talk, let alone a Rubbo, than Dina. 17 years as a postdoc. Yes, there were a few robust discussions, but they were always good. And many more years afterwards of collaboration and discussion. And I'm just delighted that this week we saw what Dina has done in her Distinguished Award Oration earlier in the week, and I was just delighted that in the same week that we've both been honoured by this society. So thank you, Dina, for that kind introduction. So before I start, I'd like to acknowledge the fact that we're on the land, the unceded land of Whadjuk people of the Noongar Nation, and I pay my respects to their elders, past and present, and also acknowledge that most of the work that I'm going to talk about was carried out on the unceded land of the Bunurong people of the Kulin Nation, and I pay my respects again to their elders, past and present. So the observant amongst you will notice there's a slightly different title than what's in the abstract. I've added the words "a personal journey," and I am going to make it a personal journey. And yes, I introduced my students and postdocs and everybody to my family, and they're going to be involved in this today as well. So let's go back in time. And first of all, let me tell you what I'm going to talk about. I'm going to give you a synopsis. I'm going to give you a historical perspective about my Clostridium perfringens journey. We've worked on a lot of anaerobes, both gram-positive and, believe it or not, gram-negative, but I'm going to focus on C. perfringens. I'm going to tell you about the development, wrong button. I'm going to tell you about the development and application of the genetics of Clostridium perfringens. I'm going to tell you about the functional analysis of a 7 PhD, 5 postdoc plasmid called PCW3, and I'm going to end up telling you about some comparative genomic analysis of Clostridium perfringens plasmids. But let's start off on the 14th of April, 1969. So a young opening batsman for Hakoah Ajax's 4th 11, an off-spin bowler, had made the decision that he wasn't going to get into the Australian cricket team. So he embarked upon a science degree and at the University of Melbourne. And on the 14th of April, I was sitting in the Woodruff Lecture Theatre, quite enthusiastic because we were about to get our first lecture from the silver-haired man that we'd seen walking around the department, Sid Rubbo. And the door opened, and unfortunately, it wasn't Sid Rubbo that walked in, it was Professor David White. And he told a shocked group of students that Sid Rubbo had passed away the night before, and there would be no lecture that day. So it's a huge honour for me to be able to give the Rubbo Oration. I didn't actually have a lecture from Sid Rubbo because that would have been the first lecture as a third-year student that day. And I guess I'm probably the last Rubbo orator that will have a personal connection to Sid Rubbo. But it's great to be able to honour him with this seminar, this talk. So from 1970 to 1975, lots of things happened. Not only on the cricket field, not much happened on the cricket field actually, it wasn't that good. That's why I was in the 4th 11. The young lad turned 21. He grew a bit of hair and graduated with a science degree, science honours degree from Melbourne University, a little later than he should have, because there were two subjects that he could never handle in his entire schooling. One was art. I'm the world's worst drawer, as my students will know. And the other was languages, and I failed science German twice, and you needed it to get an honours degree, and finally they just said, oh, get out of here, we'll pass you. And the other thing that happened from 1970 to 1975 is that I got married, grew some more hair. This is probably after a haircut. And did my first foray into human genetics. And this is young David, who's now 49 years old. So the other thing that happened is that I had a mentor, Russell Wilkinson, who passed away, unfortunately, a few years ago, and he introduced me into Clostridium, to Clostridium perfringens. So I need to tell you a little bit about this organism that has occupied most of my research career. It's a major cause of histotoxic and enterotoxic diseases of humans and animals. Diseases including gas gangrene, human food poisoning, necrotic enteritis in chickens, and other various enterotoxemias. The major virulence factors are protein toxins, most of which are extracellular. And many of these toxins are encoded on conjugative plasmids, which I'll come back to. And of course, you cannot give a Clostridium perfringens talk without some gory pictures. So here's my quota of gory Clostridium perfringens pictures. It's compulsory for C. perfringens researchers. So I think it's important that I set the scene for what was going on and what we knew about microbial genetics in 1971. And for the younger members in this audience, this is going to surprise you a little bit. So to start off with, it was primarily focused on Escherichia coli, Bacillus subtilis, and there was a little bit done on Pseudomonas aeruginosa and Staph aureus. The main focus was E. coli, and other organisms fundamentally you didn't bother with because it was too hard. The second thing, there was no recombinant DNA technology. You couldn't clone any genes. Plasmids could only be visualized by electron microscopy. There was no agarose gel electrophoresis even. There was no PCR methodology. There was no DNA sequencing. Genetic mapping was carried out in E. coli and to some extent in Pseudomonas by interrupted conjugative matings with HFR strains. I'm not going to explain what HFR strains are for those that don't know. It was complicated. Mutants could only be isolated after chemical or ultraviolet mutagenesis of whole cells, and you needed elaborate selection techniques or laborious screening to get the mutant that you wanted. And finally, there was no genomics as we now know it. You couldn't check what had happened by doing the genome sequencing. It was a very different world. But we were still able to do some research, and we were still able to get a PhD doing microbial genetics. That's important. So what was known about microbial genetics in Clostridium perfringens? This is actually a slide that I used in my first talk that I gave as a postdoc when I arrived at the University of Wisconsin. It's not actually the first slide, because that first slide would have been a 35mm slide that would have gone into a projector, and it would have been coloured on a blue background. But it explained what was known about microbial genetics in Clostridium perfringens, which was absolutely diddly squat. And we did not even know if it had a circular chromosome. So that's where we started. And so this summarizes my entire PhD, looking at the relationship between hemagglutinin and sialidase from Clostridium perfringens. We isolated mutants by chemical treatment with nitrosoguanidine. I then had to use a laborious screening method, just searching for cells that did not make this protein, which clumped red blood cells. Found out that those mutants were pleiotropic, that they were altered in sialidase production, and very fortuitously in sporulation. And then was able, because they were altered in sporulation, was able to isolate revertants by selecting for sporulating cells. And yes, in those days, it was still important to go back and find a revertant of your mutation. So that's all I'm going to talk about. I did some biochemistry as well on these proteins. Looking back at that many years later, it's pretty obvious, though we never went back and proved it, that what I'd actually isolated were mutants in a quorum sensing system, probably in the VIR-VIR-S system that we discovered some 20 years later. But that's enough of the PhD. So what happened then is that I took the family off to the United States, to the University of Wisconsin in Madison, where I had the good fortune to be selected for the slow-pitch softball team of the Food Research Institute. So here I was at Charlie Duncan's lab. I have great difficulty calling him Chuck Duncan, but Charlie, in many Charleses in America are called Chuck, and I really had difficulty calling him Chuck. But anyway, in Charlie Duncan's lab, here I am, obviously after another haircut, and this is our slow-pitch softball team, and had the highlight of that period was I had the good fortune to hit a Grand Slam home run in one match. It sounds great, but it was actually not as glorious as it was. I hit the ball to right field. The worst fielder in the other team was always in right field. It went between his legs, and I managed to get around the bases before he retrieved it. So the other thing that happened in Wisconsin is a little more human genetics, and along came the lovely Sarah. So other things did happen in Wisconsin, of course, and that was we decided to search for antibiotic resistance plasmids in C. perfringens. So I'd come in, and Duncan had appointed me because I had this microbial genetics background as far as he was concerned for Clostridium perfringens. So we used ultracentrifugation and electron microscopy to look for plasmids in C. perfringens, and we screened isolates for their ability to transfer resistance genes. And we really had some difficulty initially finding enough resistant isolates. The initial experiments weren't very successful. So Chuck had the idea that what we needed to do was get out into the to where they were using antibiotics. And so he wrote up a grant and got funded to go out to, and made some links to the pig industry. And keep in mind, we're in the Food Research Institute, so that wasn't too hard to arrange, to go out and look for resistant isolates in the pig populations in Wisconsin. So that grant got funded, and the day he got funded, he called me into his office and said, "We've got this grant, I want you to run this." I said, "Great, sounds fantastic." And he said, "I want you to go out and collect the specimens." And I thought, you've got to be joking. I don't like pigs. I don't like animals apart from little dogs. Anyway, so we had a somewhat heated discussion over a week or two, and finally he pulled rank on me and said, "Listen, lad, you'll go out and collect those specimens or you're out of here." So I went out and collected those specimens, which was one of the most important decisions in my academic career. And I'm telling you this, and I'll come back to it a little later, you can't control everything that happens in terms of your career. Some things just happen, turn out to be important, and you don't realize it at the time. Anyway, we isolated at the time the lovely plasmid PCW3, which wasn't from one of those animal isolates, but by the way. So that was the period, a very productive period at Wisconsin. At the end of it, Duncan left to industry, and I inherited the strain collection, which was a gold mine. I then moved to the John Curtin School of Medical Research at ANU, joined the biochemistry department as a gene jockey in an enzymology lab. John Morrison was tired of asking Frank Gibson's people to clone genes for him and decided to employ a microbial geneticist. And how did I get the job? I got the job, I applied for it obviously in the post with a letter. I got the job because John Morrison was visiting Wisconsin, and he called up the lab that I was in, I'd applied, and asked me to come over for an interview. I think I got the job because when he called me up, it was actually Good Friday, and I was in the lab working. And I came over in about 15 minutes, and we had a discussion, and I got the job. Again, things you can't control. We also continued our experiments in human genetics, and along came young Benjamin. So a productive 2.5 years at ANU, which, believe it or not, people don't believe, I actually worked in E. coli genetics for 2.5 years, which gave me the experience to actually manipulate genes and do the sort of work that we all do nowadays. I then moved to Murdoch University here in Perth and was here for 5 years. The experiments in human genetics stopped at this point, I might add. There's not going to be another photo of another child. That was it. The question is how did a city boy like me get a lectureship in a veterinary school? I'd never done a veterinary microbiology course in my life. So I applied for this position, got an interview, came across here to Perth. It looked great. It was a very young, very small university. And I was interviewed. It looked like research was a high priority. And I'm sitting around the boardroom table being interviewed, and the chair of the committee was the dean of the veterinary school at the time. And he asked me one key question. He said, I notice you have a publication on pigs in antibiotic-resistant strains in pigs. And I said, yes. And he said, who collected the samples for those experiments? And I said, I did. And I got the job because I knew what the rear end of a pig looked like. Later on, talking to Mel Nairn, they were concerned that they were going to get this gene jockey in who really didn't understand animals and animal diseases. And that was a relief that I answered that. Something that I had fought tooth and nail for 2 weeks not to do turned out to be critical in getting a job. So Murdoch University, I was here for 5 years. It was a very productive time. And then I moved, and that was where I set up the lab. I had that big box of strains that had sat above my desk at ANU, unopened for 2.5 years. It got shipped across to Perth and got opened, and there were all the antibiotic-resistant C. perfringens strains to work on. I then moved to Monash University in 1985, and I've been there ever since. So the next slide, I'm really going to talk about the foundation studies on PCW3. And it combines the work that was done at Murdoch and at Monash in the early days. So the first thing we did was we cloned PCW3 fragments and mapped the plasmid. And it sounds, pretty straightforward. But I actually had to write a letter to the Vice-Chancellor of Murdoch University asking the Vice-Chancellor, could he please set up a committee to regulate meat? Because at the time, Murdoch did not have a biosafety committee, and you needed a biosafety committee to clone genes from an organism that produced toxins. And so he set up a biosafety committee. He appointed me to the committee because I was the only person in the university that had ever cloned a gene. In fact, at the time, I was the only person in Western Australia that had ever cloned a gene. And then the first case, the first submission that it had was one from me, and I walked out of the room with a conflict of interest. So what we did is we demonstrated that PCW3 was the smallest representative of a family of conjugative plasmids that carried tetracycline resistance. Moving on now, that was work done here in Perth, moving on to, and really some years later, and there's a lot of other work that was done in the meantime that I'm not going to talk about. I had a visiting postdoc from Norway, Sigrid Brinstad, and she showed that an enterotoxin plasmid was conjugative and was related to PCW3 by DNA hybridization. And so we were no longer working on esoteric tetracycline resistance plasmids. We had toxin plasmids to work on. Hello, NH and MRC. So Trudy Banham, who led a lot of the early work on this project, sequenced PCW3, and it wasn't nanopore sequencing where you throw it in and get it a week later. It was cloning fragments and putting it all together with manual sequencing. It was ended up with some Illumina sequencing, and she showed it was related to toxin plasmids from C. perfringens. So again, we had this relevance issue that had fortuitously occurred. So here is PCW3, the genetic map of the plasmid. Laurie Abraham at Murdoch had mapped it to 47 kilobases by adding up fragments. It turns out he was 271 base pairs out, which wasn't bad once we'd sequenced it. We'll come back to this locus. This is the conjugation locus. We postulated this at the time and showed that this was the conjugations, the transfer Clostridium perfringens TCP locus. We have a petitioning locus here that I'll come back to. And there are the antibiotic resistance genes encoding tetracycline resistance. This was quite unusual. Those in the antibiotic resistance field called it the double because we had two tetracycline resistance genes in an operon, and that was unique and still is to my belief. Okay, so here was PCW3. We then did a lot of other work, and I'm summarizing here. I'm not even going to talk about most of this work. The work of Dina and Vicky, Isabel, Paul and Trudy on the integrative mobilizer malelement TN4451, which I must say was some of the best fun we've had, designing experiments, designing selection techniques. I'm not even going to talk about it, unfortunately. It was highly productive and a lot of fun. Just as productive and just as much fun was our studies on the regulation of toxin production in C. perfringens, done by Jackie and Sheena, who's in the room, Mike Lariskas, and in collaboration, a part of it with Kaori Otani and the late Toru Shimizu. We carried out genomic analysis with collaborators shown here, and I will mention that a little bit, including with Emily and Sam, who are in the room. And finally, we spent a lot of time looking at the analysis of antibiotic resistance determinants, work that involved Dina Liris, Dave Berryman, and others. And that was pivotal because it gave us the tools that we needed to develop C. perfringens genetics, which is what I'm going to talk about. So what were our starting points? We had cloned and sequenced erythromycin, chloramphenicol, and tetracycline resistance genes and shown that they were also expressed in E. coli, which was helpful. We had access to a multi-copy bactericid resistance plasmid from the Pasteur Institute, PIP 404. And we had the advent of an electroporator. The late Stan Falco and Lucy Tompkins came around on a lecture tour, and they were telling us about this new machine called an electroporator, and how it could be used to get DNA into cells other than E. coli, Bacillus subtilis, Staph aureus, and Pseudomonas aeruginosa. And they gave examples of other organisms. So I went and I thought, wow, we need this machine. It was at the time about $15,000. My budget didn't go that far. I went to the then head of department, Ed Westerway, and said, Ed, you've got to buy me this machine. I need it. And he said to me, okay, but tell me, if I buy you this machine, can you guarantee me that you'll get genetics going in Clostridium perfringens, that you'll be able to get DNA in? And I said to him, ridiculously honestly, absolutely not. There's no guarantee that it will work. But Ed was far-sighted and bought me the machine, which was great. It was the department's machine, not mine. And I wouldn't be here today telling the story if it didn't work. And it did. Paul Scott, a PhD student in the lab, developed an electroporation-mediated transformation system, which turned out to be very strain-dependent. Some strains you can transform, others you can't. And it's related to restriction and modification systems. And we developed a series of C. perfringens E. coli shuttle vectors. This was done by Joan Sloan, Trudy, and Dina. I'm not going to go into them, but this is just two examples. Related plasmids, one encoding chloramphenicol resistance, one encoding erythromycin resistance. So we had the tools to do genetics in C. perfringens, what we call modern genetics in C. perfringens. So we threw away the nitrosoquinidine at this point. So what have we done? Reverse genetics was now feasible in this organism. So we isolated toxin gene mutants by allelic exchange using suicide plasmids. We complemented with wild-type genes located on those shuttle plasmids, and we analysed the wild-type mutant and complemented genes in animal models. So most of you in the audience will be saying, well, yeah, okay, so that's what we do all the time. Well, this is the first time it had ever been done in the Clostridium. And we used it to look at a number of different disease situations, of two of which I'm going to talk briefly about. The first, we looked at to study the role of alpha toxin in gas gangrene or clostridial myonecrosis, gory photograph number 2. In collaboration with Rob Moore at CSIRO at the time, we looked at necrotic enteritis in chickens, and we, in collaboration with Francisco Uzal and Bruce McLean in the States, we looked at Clostridium perfringens diseases in sheep and goats, and yes, Paco did the experiments in sheep and goats, but I'm not going to talk about that. So let's talk initially about the gas gangrene study. So this was work done by Melina Awad when she was a research assistant in my lab. She's now a senior postdoc in Dina's lab, doing great things. So here you're familiar with alpha toxin, phospholipase C activity on egg yolk agar. Here's our wild type strain. Melina made 2 independent mutants. No zone of precipitation. We put the vector plasmid back into the mutant, no zone of precipitation. Here's the complemented strain with the alpha toxin gene. We restore the ability to produce alpha toxin. Now we then, at the time, we weren't doing any animal work at Monash, and we needed collaborators. And some people collaborate with people at Oxford. Some people collaborate with people at Harvard or Cambridge or Berkeley. I got to collaborate with people in Boise, Idaho. But they were great. And we set up a collaboration with Amy Bryant and Dennis Stevens. How did that happen? It happened because I met them at a meeting and met them, talked to them. They were doing stuff. I had the genetics. How about we get together? So the important message here, and I'm going to try and I've already done a couple, give a few important messages. Make sure you meet people at the meetings you go to. Go to meetings. Don't stay in your own lab group. Go and meet other people. You never know. Ask them what they're doing. You never know how useful that will be in terms of, one, making friendships, secondly, in future collaborations. So Amy and Dennis looked at these strains in mice, and very quickly, let's go back. Here's the wild-type strain, and you can see hemorrhaging and necrosis. Here's the PLC mutants. This is the dissected hind leg. We inject it into the hind leg. This is very rapid. This is all 5 or 6 hours after injection. Here's the vector, the mutant with the vector, no disease at all in any of these. Put the alpha toxin gene back in, we restore hemorrhaging and necrosis. And so what we had demonstrated, and what was thought for a long time, but there was actually absolutely no experimental evidence for, is that alpha toxin was an essential virulence factor in C. perfringens mediated gas gangrene. Sometime later, we turned our attention to necrotic enteritis in chicken in collaboration with Rubbo. So this is an important disease in the poultry industry, causes an hemorrhagic necrosis, important economically. C. perfringens is the causative agent, and alpha toxin was postulated to be the toxin that was involved in the disease. It didn't sit with me. It didn't seem right. The pathology seemed wrong. Not that I know much about pathology. And Rob agreed with that, and we had a couple of joint PhD students. The first one was Scott Sheedy, and what Scott did is he made alpha toxin mutants and put it into Rob's chicken necrotic enteritis model, and they still caused disease. So alpha toxin was not the cause of necrotic enteritis in chickens. So what was the mechanism of pathogenesis? That was the next target. So along came Anthony Keyburn, and a lot of other people were involved in collaboration on this project. We did the genome sequence of a chicken necrotic enteritis strain, and discovered, I remember John Boyce coming down and telling me, I think I found a toxin gene. It was we called it NETB, necrotic enteritis toxin B. It was encoded, cutting a lot of work short here, on a conjugative PCW3-like plasmid, which was exciting. In collaboration with Corinne Porter and James Wistock, they did the structure of NETB and showed that it was a novel beta-barrel pore-forming toxin. This is the structure of the monomeric form of NETB. Anthony Keyburn, a joint PhD student, and myself and Rob Moore made NETB mutants, and those mutants were avirulent. He complemented them, and we restored disease. And so we had shown in a chicken necrotic endomitis model that NETB was essential for virulence. This was incredibly exciting because we had overturned established dogma. And it's only twice in my entire research career, the first time was with Dina in C. difficile, that we've overturned established dogma, and it is incredibly exciting. You don't set out to do that, but when it happens, it's incredibly exciting. So that's the NETB story. Let's turn our attention to PCW3, the 7 PhD student, 5 postdoc plasmid. So here's the genetic map again, just to remind you. We had by this time worked out that a lot of toxin plasmids were very closely related to PCW3, sharing up to 35 kilobases of almost sequence identity. All of these plasmids had the TCP conjugation locus. And we called this the TCP plasmid family, and I'll come back to, there's another family now, maybe two, I'll come back to that later. And all of the plasmids that have the entire TCP locus and that we've gone ahead and tested have been shown to be conjugative. There's lots of others we haven't shown, and that's because we haven't tested them, because you need to genetically label each one to do the experiment. So this is an alignment, and PCW3 here is at the bottom. It's an easy figure alignment of some of these toxin plasmids. And fundamentally, the blue here is the TCP conjugation locus. And where you see blocks of grey, it indicates almost sequence identity between these adjacent plasmids. And you don't need to be a microbial geneticist, a genomics person to realise that these plasmids are closely related. And so what we see here is this common region here, and then other genes at the end. In PCW3, which is the smallest of these plasmids, there are the tetracycline resistance plasmid. The NETB gene is in here. Other toxins, the other end of the plasmid is variable, and it has various toxins in it, including toxins involved in enteritis in humans, and toxins involved in disease in animals, including NETB, of course, in chickens. So this gave us the incentive at this point. We knew PCW3 was the smallest plasmid in this family. It was the model system for looking at the conjugation mechanism. What are these genes doing? How do they work? What's going on here? And I'm going to cut an enormous amount of work done, I think by 5 PhD students supervised by several postdocs, down into a few lines here. And I apologise to those people for doing that, but I have no choice. It didn't give me 4 hours to talk. So what we did is the obvious thing, straightforward thing. We made mutants and tested their role in conjugation. So we made mutants, we complemented them, etc., and asked the question, if we make a mutant in gene A, does that affect the conjugation process? If it did, can we reverse it by complementation? And that work is all summarized here with the ticks and the crosses. So every gene there that has a tick has been shown to be required for efficient PCW3 transfer by genetic means. And those at the crosses are not involved. We were a little surprised about these 2 genes not being involved. This gene not being involved, that's simply a truncated version, a duplication, a truncated version of TCPA, so that wasn't a surprise. So based on that data, we also carried out protein-protein interaction studies, both genetically and biochemically, and that enables us to develop a model of the transfer complex. So this is shown here, and so it looks like, to summarize, it looks like a shortened, cut-down version of a conjugative type 4 secretion system. So we have 2 key components. We have the transfer apparatus, which I said looks like a type 4 secretion system, except there's a bit of a caveat that I'm going to throw right at the end of this, a bit of a spanner in the works. We have a relaxosome complex. The TCPM protein is the relaxase of the system that binds to the ORET site here. And we have the coupling protein, TCPA, that links the 2 components of the system. So at this stage, it just looks like a gram-positive version of a conjugation system. Sarah Revett-Mills then came on board, and she's sitting here in the audience. And again, I'm going to cut a lot of work short, and I'll come back to some other work of Sarah's a little later on. Alerted us to the fact that that wasn't the end of the conjugation genes. I'm not going to go into this work, but doing similar sorts of studies, we've got another 4 genes that are also involved in the conjugation process. And of course, there's a lot of other genes we haven't mutated yet, and it'd be lovely to mutate that, but that's going to have to be done by somebody else, not me. Okay, so moving on, here's our model of the system. And by this stage, we were collaborating again with James Wistock. And again, the message I have for you, seen with the animal work, both in chickens and humans, the advantages of collaboration. And the second major collaboration outside the field of interest of my lab was with James Wistock's structural biology laboratory. And you've seen already one result of that collaboration with the structure of NetB, but we also determined 3 structures of proteins involved in the conjugation process. And that's the TCPC protein shown here, the TCPA protein, and this is a legendary piece of work, and then TCPK, and I'm not going to talk about that at all. But what I want to do is to emphasize the insights that you can get from combining genetics and structural biology. So TCPC is required for efficient transfer of PCW3. That was shown by a PhD student in my lab, Radhika Bantwell. But it had little sequence similarity to other proteins. And when I brought James Wistock into this project, I said, look, I want you to do TCPA and TCPH and TCPF because I know they're really involved. And he said, no, I like this one because it doesn't have any similarity to anything. It's going to be new. We'll get it into a higher level journal. Typical James Wistock level of thinking. And it turned out to be a very good decision. So Corinne Porter in James's lab determined the crystal structure. And it was a bit of a surprise. It turned out it had 2 structurally identical domains. So this is the monomeric form of TCPC. It forms a trimer, and this is a model of the trimeric form. And we had shown already that TCPC interacted with itself, and all the residues we'd shown that interacted with itself were on the inside of this model, and all the residues that we'd shown interacted with other TCP proteins were on the outside of the model, so it fitted everything. But the other exciting thing was that model showed that this protein, that these domains were similar to the VB4 protein of the paradigm conjugation system for Agrobacterium and tumor fascians. And so this looked like a homologue of VB4, even though there was absolutely no sequence identity. So we think that TCPC is an assembly or stability component of the PCW3 transfer apparatus. We moved on to TCPA, and we started this a long, long time ago, and this proved to be a very difficult project. So very early on, Jenny Steen showed that TCPA was essential for the conjugation process. And data Traore, it really can only be described as a heroic piece of work, because this was an exceptionally difficult structure to solve. It took at least 6 years. He ended up having the structure of the major component of this protein in the presence and absence of ATP. And its structure related to FTSK, which has a role in cell division in most bacteria. And we knew that from limited low-level sequence identity. We'd known that since about 2006. But when the structure, when Dowda determined the structure, what we see was a very strong structural similarity, even in regions that didn't have a great deal of sequence similarity. So here's the alpha domain of the 2 proteins superimposed, TCPA in green, FTSK, the alpha domain in blue, and here's the beta domain, those 2 domains almost coincident of the 2 proteins. Now, a lot of work had been done on FTSK, and there was a cryo-EM structure. And so FTSK is involved in moving double-stranded DNA from one component of the cell to the other in cell division. And it's a hexameric ATPase, as is TCPA. And this is the structure, and this is the crystal structure with double-stranded DNA. This is the side-on structure. We knew that TCPA was hexameric, and based on the monomeric structure, we modeled the protein, and this is the model of the TCPA structure, and you see it looks very similar to the known structure of FTSK. Now and this is the side-on structure, the interesting thing is all the residues involved in the binding of FTSK to double-stranded DNA are conserved in TCPA, and that the pore size is 31 to 32 angstroms, which is large enough for double-stranded DNA to go through the pore. Now, if you read the conjugation literature, the model is always you have a relaxase which nicks the DNA and takes that, covalently binds to it and takes that single-stranded DNA molecule from the donor cell into the recipient cell. And if you look at the coupling, the relaxase of most plasmids, if you look at TRWB from R388, what you see is a pore size of 8 to 20 angstroms, only large enough for single-stranded DNA to go from donor to recipient. So we float the possibility, and this is work that was published this year, we float the possibility that in C. perfringens what's happening is that double-stranded DNA is going from the donor to the recipient. Do we have the evidence for that? No. It's actually a very difficult experiment to do in organisms other than E. coli. It can be done and should be done. We simply float that hypothesis. Okay, coming back to Sarah Rever-Mills, she's done some, and this is the only slide I'm going to show you that hasn't yet been published, so I'd ask you please not to photograph this slide and tweet it. So Sarah used super resolution microscopy, or STED technology, to look at the location of these proteins, and I'll summarise again a lot of work in one slide. Sorry, Sarah. So what she did is she looked at TCPM. So we tagged these proteins, showed that they were functional, and looked at them under super resolution microscopy. So the relaxase protein, TCPM, is located in punctate foci all throughout the cytoplasm of the cell. When she looked at TCPA, the coupling protein, a different result. This protein appears to be associated with the cell membrane, and that's no surprise. It has two N-terminal transmembrane domains. Not shown here is the fact that two small transmembrane proteins, TCDD and TCDE, also appear to be located in the cell membrane. When Sarah looked at TCPH, she got a result that said that it's primarily located at the cell poles, and again appears to be membrane associated. So all of these data are totally consistent with our model for the conjugation process. Okay, let's move on to the last part of this talk and talking about the comparative genomic analysis of C. perfringens plasmids. And I'm going to talk about a problem. So we had a plasmid compatibility problem. So we noticed very early on that many C. perfringens strains had more than one plasmid. Well, so what? We've seen examples at this meeting. People have talked about strains with more than one plasmid. But these were all PCW3-like plasmids that had 20, 30 kilobases of sequence identity. They shouldn't all be there in the same strain. So one example of that is the necrotic enteritis strain. I think I've got ahead of my slide collection, EHNE18, which that earlier work had done. All these toxin plasmids had closely related replication, maintenance, and conjugation loci. How can they be there in the same cell? So this is the example from NE18. 3 plasmids, all conjugative, independently conjugative, a tetracycline resistance plasmid, a beta-2 toxin plasmid, a NET-B toxin plasmid. All this region from here to here is almost identical. Why are they there in the same cell? They shouldn't be compatible. What's the basis for this? Enter Vicky Adams and Tom Watts, and Vicky's online and Tom's in the audience. We looked at this problem. So when we look at the replication region and we look at the rep protein of these plasmids shown here, they're closely related, 98 to 99% identity. But when Vicky looked upstream, there was a partitioning locus, a PAR locus, where the level of sequence identity was much lower. And so what does this locus do? To cut a long story short, in these types of things, this is not restricted to C. perfringens, they're in lots of different bacteria. The PAR-R protein encoded by this gene here binds to this locus, as shown here. That then brings the PAR-M protein to the complex. That polymerizes and pushes the replicating plasmid molecules to the opposite ends of the cell, so that when cell division occurs, the daughter cells inherit both plasmids. So it appeared that these plasmids had different partitioning loci. So Tom Watts looked at this and showed that plasmids with the same PAR-MRC loci were compatible, and plasmids were incompatible, and plasmids with different PAR-MRC loci were incompatible. And that's let me start that again. Drink of water time. See, Mark, you're not the only one that can stuff up from the front podium. So plasmids, I'll read this to make sure I get it right. Plasmids with the same PAR-MRC systems are incompatible. Plasmids with different systems, like the ones I showed you before, are compatible. He went on and with the assistance of data Traore, showed with surface plasmon resonance that the PAR-R proteins interact with cognate PAR-C sites, but not non-cognate PAR-C sites, that showed that the PAR-MRC systems are primary determinants of toxin compatibility or incompatibility in C. perfringens. So we can understand, and our record, I think, is 6 or 7 PCW3-like or TCP plasmids in the same cell, all with different PAR-MRC loci. Okay, genomic analysis of C. perfringens was initially done by Toru Shimizu in 2002. We collaborated, and the work was primarily done by Gary Myers and Ian Paulson in the States before they came back to Australia. We did some other sequences, but more recently we've had a fantastic collaboration with Emily Gulliver and Sam Forster, who are also in the room, and they've looked at the analysis of 453 genome sequences. So we've come a long way from that circle with the question mark in the middle, of which 104 were sequenced by Emily and Sam. 5 distinct genomic clades were recognised, but these were not related to the 7 known C. perfringens toxinotypes. So I haven't gone into this, but perfringens has 7 toxinotypes. And those toxinotypes we now know are plasmid determined, primarily determined by the presence of a plasmid carrying a toxin gene. So these are this is from Emily's work. These are the 5 genomic sequence clades. Clades 1, the major clade, 2, 3, 4, and 5, not related in any way to antibiotic resistance genes, to toxin genes, or mobile element genes. They're spread around, or they're spread right through all the clades. So the genome clades are not related to the way we tend to type C. perfringens, which is toxinotyping. We then went, Vicky and Emily then went and looked at the plasmids, and we decided that we needed some closed plasmids, and we ended up with 56 closed plasmids from 11 different isolates from all 7 toxinotypes. And we, again, cutting a lot of work down to a short summary, we found there were 6 different groups of plasmids. The predominant groups in these strains were the TCP plasmids. There were 24 of these that were related to PCW3. They were conjugative, they had toxin and resistance genes, they had the TCP locus. There were 9 plasmids that had another conjugation locus, and this was the PCP locus. And I haven't gone into this, but there's another set of conjugation genes in C. perfringens, the PCP locus, first identified in another Clostridium in Dina's group, and in collaborating with Dina's group, Tom Watts showed that PCP13, one of these plasmids, was conjugative. Interestingly, we've come across what may be the 3rd conjugation locus in C. perfringens. We have 2 plasmids, which we call the BCP locus, because it seems to be related to plasmids in Bacillus anthracis. Potentially conjugative, we don't know. And then we have some other plasmids I'm not going to go into. So it's much more complex than we thought. And this simply summarizes the TCP and PCP plasmid families. I won't go into detail, simply remind you in these Brigg diagrams where you see bars of colour. Each bar is a different plasmid. When you see bars of colour being common, that means there's sequence identity, and that includes the conjugation loci, both in PCW3-like plasmids and in PCP13, the PCP-like plasmids. And it's these regions here that are different that have the antibiotic resistance and toxin genes. Okay, so in conclusion, what have we shown with all of this journey through Clostridium perfringens? I hope I've shown you that the development of C. perfringens genetics has led to significant advances in our understanding of the pathogenesis of C. perfringens mediated diseases. It's also enabled us to branch out into other Clostridia. A lot of that work has been done brilliantly by Dina's group, and I've not talked about any of that. Secondly, the genetic, functional and structural analysis of PCW3 has led to the elucidation of a model for the conjugative transfer of this important family of toxin and resistance plasmids in C. perfringens. And thirdly, comparative genomic analysis has led to a detailed understanding of the nexus, and that's why I included it in the title, between conjugative toxin plasmids and the toxinotyping, the typing of C. perfringens isolates. Now, surprise, surprise, this has not been all of my own work. And here's a long list of all of the people that have been involved. And I must apologise, Sasha, wherever you are. I left your name off the list, sorry. And there's probably somebody else that I've left off the list, and if they're here, I apologise. If they're not here, I also apologise. I'm not going to go through all of the names. These are the major PhD students involved in the latter work on PCW3. These involved in the earlier work and in the work on developing the genetics. And I can't help but congratulate Priscilla, who was involved in some of the tetracycline resistance work, on becoming a professor this week. Well done, Priscilla. I must thank, in particular, Vicky, Trudy, and Dina, who have led this work, supervised most of these students. They've been fabulous postdocs to have in my lab. The 17 years that Dina and I worked on these various projects was exciting, and it's been really exciting for me to see what she has done when she's established her own group and has gone on to bigger and much better things. So well done, Dina. I must thank, and there's a group of people that have been totally left out, and I've put their names up because there's another project that I've worked on for nearly 40 years, and that's the gram-negative bacterium Dichelobacter nodosus that causes foot rot in sheep. And I've not said anything about that, and they always get annoyed because I don't mention that work when I give clostridial talks. But here are all the people that were involved in that project, including the first ever Monash student that worked on that project, Marina, who's in the audience. And I just need to thank them for their involvement. They did input into the Clostridium perfringens projects, and for most of that work, that was led by Ruth Cannon. I also need to thank the collaborators that were involved in these projects from various institutions, Dina's group for her independent group, over the last dozen years or so, James Wistock's structural biology group, the more recent collaboration with Emily and Sam, the long-term collaboration with Bruce and Paco on the animal toxinotype strains, another really productive collaboration with Rob, and various other people in various other places that we've collaborated with. And again, that take-home message: go to meetings, meet people, ask people what they do, and say, gee, could I collaborate with this person? Can they do something I can't do at the moment? So I also need to thank the sponsors of the work, the people that we obtained funding from over the years. I'm not going to go through all of the funding sources, both the granting bodies and the home institutions that funded the work. There's one other piece of thanks that I must have. This has not been a solo journey. It's been a 50-odd year journey, but I had another person with me for most of that journey. This is going to be difficult. I first met my wife when I was an 18-year-old second-year student. She saw me go through an honours degree. She saw me go through a PhD, supported me for all of that time, through all the travels overseas, taking her to places she didn't really want to go and live. And I need to thank her. Unfortunately, she's not here to listen to this lecture. She would be immensely proud that she's listened to Rubbo orations. She knows many of the people in this room. And she would be immensely proud that I'm up here giving this talk. And I owe an enormous amount to her for supporting me all the way through this very personal journey. I also need to thank this bunch of reprobates. They grew up, the little ones that I showed you earlier, and this is they grew up into amazing adults, and here they are with their wives. And there's one other person I must thank, and that's my nearly 102-year-old mother, who, again, supported me right through my life, pushed me to study when I was a young child, and has been incredibly supportive in everything that I've done. So that's family. This is also family who have the ability to bring some chaos into my life, joyous chaos. And so this is the end result of that practical human genetics. 3 has gone to 7. But there's another family that I must thank, and I thank them immensely, and that's this family. I joined ASM as an honours student in 1970. I went on to the branch committee, the Victorian branch committee, as a hot-headed PhD student, fired up that ASM wasn't doing enough for PhD students. I served on the ACT branch committee as secretary. I served on the WA branch committee for a short while. I've had a role in the national body as vice president, as president, and have had a role on the NEQB for the last half dozen years. But the most important thing is that it has been a fantastic society that has supported me. The friendships that I have gained through my involvement of this society have been immense. I am incredibly grateful to this society, yes, for honouring me by awarding me or asking me to give the Rubbo Oration, but I'm more grateful for the society for what it's done for me and the opportunities it's given me to interact with all of you and all of the people that I've interacted with over those 50 odd years. Thank you, ASM. Thanks a lot.